It's great to be here. My name is Britta Belli. I'm head of PR for In Silico Medicine. We're a global AI drug discovery company. Before I was at In Silico, I spent many years in communications at Yale, and I was here for the first Yale Innovation Summit. So it's such an honor to be here for the 10th Yale Innovation Summit. And it's also an honor to welcome Dr. Matt Truppo, Global Head of Research Platforms and Computational R&D at Sanofi. Dr. Truppo oversees the full range of research platform capabilities at Sanofi, including integrated drug discovery, large molecule research, drug metabolism and pharmacokinetics, translational models, and preclinical safety. He is also responsible for driving R&D's digital transformation roadmap to optimize and accelerate R&D processes leading from target identification to approved medicine. Dr. Truppo's experience in the pharma industry spans a variety of roles across drug discovery and development in both the small molecule and large molecule arenas. He was previously Vice President of Johnson & Johnson, leading protein therapeutics, API development, and Vice President of Biologics Discovery at Johnson & Johnson. Before J&J, Matt spent 17 years conducting pharma research and development at Merck. He received his BSc degrees in chemical engineering and engineering biology from Princeton, and his PhD in chemistry from the University of Manchester, working at the Manchester Institute of Biotechnology. Dr. Truppo has served on multiple international scientific advisory boards and has authored more than 50 publications and patents. He is a fellow of the Royal Society of Chemistry and a member of the American Institute of Chemical Engineers and the American Chemical Society. Please join me in welcoming Dr. Matt Truppo to the stage.
So thank you so much, Britta. Can folks hear me all right? Good, no? There we go, it's coming up. So thank you very much for the nice introduction. It's a pleasure to be here and be invited to speak to you all today. I'm going to talk to you a little bit about AI applications at Sanofi, particularly across R&D. And it's exciting to give this talk, exciting to give it to all of you. And I've heard there were several conferences and talks yesterday that were exciting, thought-provoking. Hopefully we'll get into some of that in the Q&A afterwards as well. So at Sanofi, our goal is to become the first large pharmaceutical company powered by AI at scale. And what does that mean to us? What that means is that we can apply, develop, and deploy AI tools to the thousands of Sanofians around the world in order to develop better science, in order to improve our productivity, and in order to make better decisions that are data-driven in a more rapid and informed way. And we're doing this with 3 types of artificial intelligence. And I put them up here. And the first one, I think, was coined by our CEO, Paul Hudson, snackable AI. So snackable AI, or it's AI that we can deploy rapidly to thousands of people in their pocket that allows you to make better decisions on a daily basis using very large data sets and ingestion of very large data points. And then we have expert AI. This is applications of massive compute, the type of things you would classically think of as AI, massive computing power, massive data sets with subject matter experts to solve very complex problems. And then we have generative AI, where our initial forays in general AI are in document generation to take very complex, time-consuming, but critically important work out of the hands of folks and be able to free up their time and be able to have higher quality document generation. So I'm going to talk a little bit about that as well. So what I'll do over the course of the next 20 minutes or so is talk about each of these applications of AI. I'll talk about applications that are in use today, so things that are deployed already over the last 6 months, in use across our network, in use by thousands of people, but also give some insights into what does the future look like, what do the next 12 and 24 months look like at Sanofi. So first, snackable AI. Now, where we've applied this snackable AI concept first is in project and portfolio management. This is an area where we've worked with Ailey Labs to develop a play app, which I'll show you in a second, which is on the phones of, again, thousands of people across the company. And what this does is collect billions of data points from both internal inside the company as well as external competitor data points about our portfolio, about external portfolios, about our clinical trials, other clinical trials, and general information in the healthcare sector. What that allows it to do is in one app make predictions and portfolio simulations in real time based on data that's getting pulled in on a daily basis. The app then automatically makes recommendations about what you might want to do, not just across the strategy of your overall portfolio makeup, but also within each individual project. And I'll show you an example of that. So this is the app. So if I pull out my work phone and go to the Play app, you'll see something like this on there. There are multiple applications. I'm going to focus on one now, and I'll show you one other one later, but we have 4 or 5 of these at this point deployed. So in this portfolio management app, you can see if you start all the way on the left-hand side, that's an image of the net present value calculated for our R&D portfolio by therapeutic area, okay? As you come in each day, anything new that's happened internally or in the external world will be highlighted automatically on your phone. And it's in reference to your specific job function so that you can take a look at things of interest to you. It will also make recommendations at the portfolio level and the asset level. So from a portfolio level, it might say, you know, what if you unwound and had capital reallocation from this project to this project? That would probably increase your net present value of your portfolio overall. It'll make these suggestions based on the data it's ingesting. That may be a successful clinical trial that's run by a competitor, which now puts into question how competitive is our asset, or vice versa, a competitor's clinical trial that failed, now putting us in the lead. And it will automatically make those suggestions for what you might adjust or change. So it doesn't take the decisions, though, out of the hands of the human being. We are still responsible for those ultimate decisions. What it's doing is prompting a conversation about return on investment on a project and portfolio level. So then if you move to the right-hand side, each of those individuals can then go into that app and say, let's run some what-if scenarios. Let's say that we do reallocate capital from project X to project Y, and we accelerate clinical trial enrollment as a result of that. How does that change the overall portfolio mix? And you can see that clinical trial enrollment increasing. You can see the clinical trial, how early it might read out as a result of that change, and then the return on investment for that particular project change, which then adjusts the NPV of your entire portfolio makeup. So you can do this again in real time based on current and up-to-date information. So we've used this to make decisions over the last few years. We've used this to inform our decisions. And we have on real projects, in some cases, stopped those programs, or in some cases, accelerated other programs. What that has led to over the last several years is a pipeline value that's about 4-fold higher compared to 2019 of our research portfolio. Now, this alone, a portfolio management tool, is not sufficient to deliver that sort of value chain. These projects have to come from somewhere. They can't just be prioritized. They have to come from somewhere. And that's where we lead into expert applications for a truly efficient, high efficiency R&D engine feeding projects into the pipeline. So we'll talk about and move over to some expert AI applications now. I'm going to talk about these applications across the whole value chain of R&D. So starting with understanding of disease biology, then moving into the drug invention space, we're actually designing the drug we want to test. Then we move into clinical trials, look for safety and efficacy, and then ultimately, how do we supply that drug with a commercial manufacturing route that can treat every patient in the world that needs it? And so I won't go through every one of these areas, but I will focus on several of them, and I'll highlight 2 things. One is, what tools are we using today that have been developed over the last 6 months to a year and have been deployed, as well as what does the future look like that's going to be developed over the next 6 to 12 months. I should also say that we don't do this alone. We have friends. And you can see a lot of partnerships on the bottom of this slide where we're working with subject matter experts that have complementary skill sets to our own, where both companies benefit in some way. And we can talk about that a little bit later if folks are interested as well. All right, so the first expert AI application, I'm going to start at the beginning of our process. So we're going to walk through when a target enters the portfolio all the way through to filing for launch. So target identification is the start of our process. It's the start of our journey in R&D. We want to identify a biology that's impacted by a target so that we can either mitigate, eliminate, or cure a disease. What our teams in the precision medicine and computational biology group have done is develop target ID engines. These engines initially focused on core areas of expertise to Sanofi in the immunoscience space, and key critical disease areas that are core to our immunoscience portfolio. And you see some of them listed here on the left: atopic dermatitis, ulcerative colitis, asthma, COPD, and others. These target ID engines combine multimodal data sets of public and private information. They put it through a machine learning model that we developed in-house, and it rank lists and orders targets of interest against those diseases. We then filter those through other models based on 3 factors: druggability, safety of that target in biology, and the competitive space to make sure that we can actually win. Those then top hits are then analyzed to see if we add them to our portfolio. So the exciting thing about this is this was developed over the course of maybe 2 years. In 2013, the first year where this was actually deployed widely, we were able to evaluate 50 target hypotheses with a relatively small group and put 7 novel targets into the pipeline in 2023 alone. So a really tremendous effort for the initial foray into this space. And we're now expanding this to other disease areas as well as other therapeutic areas like neuroscience and oncology and others. So now we have a target we want to go after. The next step is inventing the drug. The drug invention process is a really iterative, time-consuming cycle. It can easily take 3 to 4 years at best. It can encompass a variety of modalities, from small molecules made with synthetic chemistry to large molecules like biologics, antibodies, nanobodies, cell and gene therapy, for example. It's also a space where you're combining now true subject matter expertise across a very wide range of disciplines. So bringing all this together in an easy way and in a fast way to deliver quality molecules is very difficult and challenging. And what I'm going to talk through is some examples in both small molecule synthetic chemistry space as well as large molecule biologic space to show you what we're doing a little bit here. So the small molecule space, one of the things we're always interested in is how do we increase the quality of our molecules and increase the speed with which we can deliver them. So one aspect that we work on is explainable AI for small molecule drug design. So over the last couple of years, we have found that there have been AI models we've been able to develop internally and with external partners that have been getting highly predictive of certain properties of small molecule drugs. An example on the left-hand side here, you see all these green dots. Each one represents a synthesized small molecule against a target. It shows the biological activity, so experimental calculation on the x-axis and predicted by the AI on the y-axis. And this is a real program. In this particular model, you can see it's quite good. If you're perfectly on that diagonal line, it's a perfect correlation. It's over 80% predictive. So that's great, and we can combine these tools with our other more classical approaches of structure-guided drug design, of methods using statistics, of physics-based methods to be able to inform on drug discovery. But one of the challenges with AI models is they can be a bit of a black box sometimes, and in order to combine the learnings from them with all of the other tools at our disposal, we need to make it a bit more explainable. So we've used a statistical game theory analysis, a SHAP analysis, to be able to do that. And in simple terms, because I am not an AI expert, this is not my background, but in simple terms, I'll focus on that center picture and show you exactly what we're doing. So that big blob around the outside is the protein structure that we're docking the drug in. In the center, in the ball and stick model, is the actual drug itself. The system will go back and take a look at what the AI has done and start to highlight regions of that molecule in green and red. In this case, which portions of the molecule in green are quite malleable into new design that can help with biological activity, although you can imagine any factor or specific parameter you want, and which portions in red need to be conserved, because as they're changed, it actually makes it less biologically active. When we then dock that into the protein of interest using standard structure-guided techniques, we can start to learn, why is this molecule being changed by the AI in this way? How is it affecting the actual biological function? And that allows us to combine information from the AI with information from all of the other approaches that we use. And then we don't stop there. We then port this over into a VR environment. So you can see one of our chemists with a set of VR goggles there. And this is not a feature or gimmick. This is used every day in our labs. We have several of these labs set up across sites where the scientists can go and shrink themselves down to the size of a molecule in a virtual environment and actually see how that drug interacts with the protein target, and in real time do calculations and molecular changes and simulations in the VR environment. And more importantly, and what's powered up even more by this VR environment, is the ability to collaborate across sites. So we're able to take folks, our chemists in Germany, in France, in the US, and simultaneously collaborate in a virtual room on the same molecule. And in this way, you can generate ideas with a diverse set of opinions, a diverse set of talent that's separated by geography in a very efficient way. So we use this routinely on projects, and it's actually quite fun to stick yourself in the virtual room and see and understand things from the perspective of the molecule. Okay, so that's a little bit about what we're doing today and has been deployed and used. I should mention, and I think it was on there, but I didn't say it, we're now using AI tools as part of our workflow in 75% of all small molecule discovery programs. So they are getting to be pretty widely adopted, and this is fairly new over the last 24 months. But what does the future look like? So this is a capital-intensive project that we just kicked off, and it's what the future of drug discovery looks like in the small molecule space for Sanofi. If you think of the cycle that we use to invent small molecules, it's a design, make, test, analyze cycle. So you design the drug using the tools that I just mentioned before. You're then going to make and synthesize that using synthetic chemistry. You're then going to test it in biological assays, you analyze the results, and then in this virtuous cycle, you go back again, and you inform yourself on the next stage of design and go through that cycle again. And this can be done dozens of times until you arrive at a suitable and acceptable preclinical candidate. There's a lot of white space in this process. There's shipping of compounds. Many of these components are not connected. So the idea is to connect and deploy a closed loop design, make, test cycle, fully automated in a single lab. We're building this in our Frankfurt site, and some of these components already exist today. We already have high throughput chemistry and synthesis. We already have high throughput biological assays. We already have many of the design tools that I just mentioned and showed. So what we're going to be doing is stitching these together in such a way that both the scheduling software for the automation, as well as handing off from one robot to the next, as well as the critical factor, which you see this line connecting the analyze to the design, the analysis will feed directly back into the machine learning algorithm for the next round of design, which will start on the robot without any human intervention. That's the piece that really takes quite a bit of time to come in the next morning, do that analysis, run those models. If we can stitch together a protocol that then allows us to do this in one iterative cycle, you can imagine accelerating. And what we've seen is removing that white space will lead to a 50 to 80% reduction in that DMTA cycle. Now, practically, this won't be able to be applied to every program. Not everything can be done on the same deck because of capabilities of automation for different types of chemistry, but we have done an analysis of all of the chemistry that we use across all our programs, and we think about 60% of it is amenable to a complete closed-loop design, make, test cycle, and the rest we can fit in with scheduling software, with some of it being done off deck and some of it being done on deck. So now I'm going to move to large molecules and give an example in the protein design space of the application of expert AI. So mRNA has been, ever since particularly the outbreak of COVID, on many folks' minds, and mRNA drugs have really transformed the industry in terms of vaccines and beyond. Our mRNA Center of Excellence has been working on a large language model to predict properties of mRNA to enhance and aid in mRNA drug design. This is an example from our mRNA flu vaccine efforts. So we developed a language model called codon-BERT that looks at the codon level encoding of proteins for mRNA. This has been trained on 10 million mRNA sequences, and what we've been able to show is that it is indeed superior to all other large language models that we've seen for prediction of mRNA properties, in particular expression in vivo of that protein the mRNA encodes for. What that leads to, it's about a 10% increase compared to other large language models that we've studied. And what that leads to is a lower dose, higher efficacy, potentially increased safety, and lower cost of goods. So we've applied this to our flu programs in mRNA, and it's now being applied more broadly across all the mRNA programs in our mRNA Center of Excellence. So again, example of something we're doing today. What we're doing in the future, this is probably my favorite project. Don't tell all the other groups. This is probably my favorite project at the company in terms of applications of AI. This is our Biologics AI Moonshot initiative. BioAIM. It is a collaboration between our large molecule research folks that do biologics discovery, our digital teams, and an external partner, Biomap, which has a 100 billion scale protein language model. So protein language model, as you may know, is kind of a corollary to large language models. Large language models are tremendously impactful in terms of understanding human text and human language. And with the 26 letters in our alphabet leading to all of the words and sentences and paragraphs that we use to describe things in the English language, you can understand that the 20 amino acids in proteins could possibly be a language that we could crack the code of and understand as well. And that language and that order gives rise to the primary structure, the tertiary structure, the three-dimensional functionality, and all the biophysical properties of that molecule, if only we can understand it. So the goal here is to develop large language models for proteins and refine them based on data sets that we have of highly curated antibody and nanobody data sets with millions of antibodies, millions of nanobodies bound to the receptors with all the target information, all done in exactly the same way. So we have very highly curated data sets, which was something attractive to Biomap. Biomap has the largest large language or protein language model in the protein space, very attractive to us, so it was a really good partnership. And I'll describe what the goal is here. So if you start with how do we do this today in terms of state of the art for, let's say, an antibody discovery program, you would immunize a transgenic mouse. You would let their immune system and hypersomatic mutations generate hundreds of thousands to millions of antibodies in response to that antigen. And then you would sort through all of those to find ones that bind to the target you're interested in. After that, you would isolate, purify, express all of those, and then you would test them, validate them, and now you move into engineering to make them drug-like, to improve their druggability properties for formulation, for stability, that sort of thing. And this, again, is an iterative cycle, and it takes quite a bit of time. If you can have protein language models that predict those properties like which epitope do they have, what affinity will they have, how developable will they be, then you can start to, in silico, down-select most of those sequences which would be detrimental and up-select the sequences which would be beneficial. So you can see how you can shorten the time cycle by having to physically make far fewer things and physically test far fewer things. So that's a slot into what we do today, and we will be deploying that AI-guided hit selection tool by the end of this year. But then you can think about the future of eventual de novo design. Could you actually do this de novo with a target and build up that amino acid sequence using diffusion models and other things demonstrated by the Baker Lab? And I do think that that's going to be possible at some point. Right now, it's not possible to do all the parameters that you want to build into a drug, but I do think that in the coming years, this could be a direction where you bypass the need for the animal completely. So you can see, and this is an example where we're building the tools to be able to address today's needs, tomorrow's needs, while building toward a future that is even more transformative, delivering value all along the way. All right, so we've moved from inventing the drug. Now we want to test it in human beings. We want to look for safety. We want to look for efficacy. So we're going into the clinical trial optimization space. This is a space, running clinical trials, where it is the most time-consuming and the most expensive part of what we do. It can easily take longer than 5 years. It can easily cost upwards of $1 billion. So anything we do in this space will dramatically affect the cost of R&D in the pharmaceutical industry. So we're doing actually a lot in the clinical trial space. I'm just going to show 2 examples. The first is in the site selection of trial sites in terms of hospitals where we're actually going to run this trial. And the way this was previously done is many different data sources, and I hope you're seeing a theme here of many different data sources, multimodal data. This is one of the true hallmarks of where you can leverage AI. Many different data sources coming from all different places and all different types had to be pulled together manually. And that process was very time and labor intensive. On top of that, you can get bias in the system for individuals that had a good or bad experience running at a particular site on their particular project in the past. So not a great way to decide and pick clinical trial sites. To a system now where we have an AI-guided selection tool for clinical trial sites that brings all this data together in real time. And I'll show in a second that this is also now on the Play app, so I'll show you that in a minute. And we can optimize those trials for speed of patient enrollment, for cost of the study at different sites, and for the diversity of patients that we want to include in those trial sites. And so the proof is in the pudding. So if you see at the bottom here, we've been able to compress the timeline to validate sites from multiple weeks to 4 days. The sites that have been selected with this tool compared to the standard way has a 1.6 times faster enrollment. And that leads to a projection of approximately a 4-month decrease in the clinical trial time. And if you imagine a 4-month decrease in clinical trial time leading to a 4-month faster registration and 4 months faster in the hands of patients, it has an impact on patients, it has an impact on the economics. So tremendously powerful, and I'll move to where we haven't stopped there. So we also have data-driven monitoring at the sites where we send out monitors from Sanofi to ensure that data quality, to ensure everything's progressing smoothly at each of our sites. And the way this was done in the past was really a one-size-fits-all. We'll send people at a regular frequency to each site to ensure everything's going well. Now that we have a system that pulls all of this information together in one place, we can behave a bit differently. And we can do things that are more fit for each site. We can take a look at simultaneously the site activities that are projected to occur over the next year, the pace of enrollment, how many patients are already enrolled, as well as the volume of data that's coming out of that site. And we can put the monitors in the right place at the right time whenever they're needed. And what this has led to is our data monitoring group improving the data quality. It's also led to a 30% improvement in productivity for that team with respect to where they need to be at the right time, being prompted by the AI and saying this is the schedule that you should adopt based on the data coming out of each site. I mentioned that this is also on the Play app. So again, if I take out my phone and go to now the ClinOps and Clinical Control Tower app, so I've shown you two now, the Project Portfolio Management app. This is now the Clinical Control Tower app. We have several others. There's a finance app where the entire finances of the company, again, in one place. It is restricted based on the user security settings. So you can see what you are allowed to see. You can see what you need to see to do your job. But it's quite powerful to have this in one place. It's actually changed the way we do meetings. We pull out the Play app either on our phone or on the screen rather than having PowerPoint presentations. And that's intentional. We want folks to be able to adopt and use this. So the Clinical Control Tower app's been rolled out to several thousand users. It's one single source of truth for data, which is always very helpful in conversations. And you can see we can run reports, we can run analytics, and we can do the simulations similar to what I showed before in the portfolio management. We can do simulations in terms of clinical trials as well. So the last example of AI application I'm going to give is in the generative AI space. And I mentioned this type of AI we're first starting in document generation as a way to alleviate the burden of very onerous but really critically important activities that need to occur. So we've now identified our target, we've invented our drug, we've tested it in human beings in clinical trials, and it's time to start submitting to the regulatory authorities. So where we started was the reports that are written called clinical study reports, or CSRs. These CSRs are comprised of the activities that were conducted in the clinical trial as well as the results, and they get submitted as part of the package for filings to regulatory and health authorities. They're very, very long documents. They're hundreds of pages of long. We do a lot of them a year. We do about 50 or more a year, and that's going to increase as our really promising late-stage pipeline at Sanofi. We anticipate 50% more phase 3 clinical trials in the next 24 months, so this work will only increase dramatically. It takes about 36 weeks to create each CSR. So a very labor-intensive document, a very technical document. The goal was to, in 6 months, develop and deploy a clinical study report engine that could deliver on the first few sections of the report. And the sections we focused on were section 3, the study design, and section 6, the conclusions. Those represent about 35% of the total technical complexity of the report, and it was kind of our proof of concept of would this work. So I'm happy to say that this was delivered 2 weeks ago and deployed to all clinical study report writers at Sanofi. And we're going to write a report right now, so you can see how fast it is. All right, so you go to the My CSR page, very aptly named, and click Generate New Report. You select the molecule, the drug you're interested in, as well as the specific trial, and you say, I want to create a CSR. On this page, you're now selecting the primary source data. So the study protocol, the results, and all of the other information that includes thousands of pages of documentation that's been collected at all the sites. From this information and data, you'll now go next, and you can see here's a CSR, and here's our first trial for section 3, the investigational plan. That's what we have in place. And we're now going to generate it, and it's done. So now we're going to click on View. And here's the clinical study report, starting with the investigational plan and study design, the dosing regimen, and all of the other subsections of CSR section 3. So the end result of this first case deployment has shown that we've been able to create a 70% complete document in less than 10 minutes of clinical study report writers' time. This has saved weeks of writing time off of this portion of the document alone. And this has been successful enough that we're now going to deploy it to the other sections of the CSR and start making modules for those, as well as beyond the CSR to the entire regulatory filing, which, if you're not aware, is about 100,000 pages long. So you can imagine experts writing 100,000 pages of documentation, pulling from millions of pages of source data. This is a perfect use case for generative AI, and we can focus our experts on that final 30%, or maybe less. We're trying to get it to 80%, that final 20%, to ensure that the quality, that the formatting, that everything is correct. Interestingly, and I cut the video off short so we could move on, but you can also make suggestions and modifications and changes to the output from that generative AI tool. And those suggestions and modifications go back into the learning of the AI so that it can learn how to make a better CSR in the future. So it is a loop in terms of learning what the actual writer wants to deliver. So with that, I hope I showed you a little bit of a glimpse into the entire space across R&D, from understanding of biology to drug design to clinical trials, and also to filing and document generation, of how we're applying AI tools today at Sanofi, a little bit of a glimpse into the next 12 months and what it might hold. And we really believe that AI plus human beats human alone. AI is not meant to displace human beings. AI is meant to help us take very large sources of information, very large disparate sources of information, and inform our decisions better, inform our decisions faster, so that we can create medicines to help and improve patients' lives in a much more profound, cost-effective way. And with that, I'm going to stop, and we can move to the next part of the section.
Thank you, Matt, so much. This was such a fascinating presentation, and we're now going to get to dig into the details a little bit more in a fireside chat with my colleague, Patrina Kamya. Patrina is the global head of AI platforms and president of In Silico Medicine Canada. She oversees In Silico's AI platform, Pharma AI, which includes novel target discovery, novel small molecule generation, and clinical trial outcomes prediction. During her PhD at Concordia University in theoretical chemistry, Dr. Kamya learned to change the functions of molecules by manipulating their structures using computational algorithms. She later consulted for pharma companies at Certara and led sales and business development of molecular modeling software for pharma and biotech companies at Chemical Computing Group. Then she discovered the advances happening in AI drug discovery and was immediately invested. At In Silico's R&D Center in Montreal, Dr. Kamya leads a dedicated team that not only advances the software platform, but collaborates with academic partners at University of Toronto, incorporating AlphaFold and quantum computing to identify new targets and potential therapies for cancer and other diseases. Dr. Kamya has been profiled in Chemistry World, interviewed in the Globe and Mail, and was featured on the Nova PBS documentary, AI Revolution. She's presented on numerous conferences on the rise and potential of AI drug discovery, and is leading In Silico's efforts to use AI to design non-hormonal contraceptive options for global women in need, with support from the Bill and Melinda Gates Foundation. Please join me in welcoming Dr. Patrina Kamya.
Thank you very much, Matt, for a very inspiring keynote. Thank you very much, Yale Ventures, for inviting us to have this fireside chat. I think we're at a point in time in the adoption of innovation in drug discovery and development where hearing stories like yours are very, very important in terms of understanding the impact that AI can have in our space. Thank you, Britta, as well, for the introduction. That was great. So what I'd like to do is spend the next 20 or so minutes picking your brain on three topics related to innovation in pharma, and specifically the role that you play. So I'll start with your journey. I think that many people would appreciate learning how you got to where you are. I mean, not too many people know that you're from New Jersey, not too far from here, and I think your career is very, very impressive. And then the second topic I'd like to discuss with you is touches on some of the challenges associated with adopting innovation in big pharma in particular. And then lastly, I was going to ask you about the successes, but you've done such a great job talking about that. So I'd like to pivot and maybe talk more about the risks and how you're mitigating the risks associated with adopting AI in drug discovery and development in particular, and how you're thinking about responsible AI.
Sounds great.
Okay, great. Let's get started. So I met you a few months ago, Matt, and when I met you, I was very, very intrigued about your journey, and I think that a lot of people would appreciate learning more about that. So why don't you start? You were born in New Jersey?
Yeah, sure. So I was born and raised in New Jersey. It is a good state, despite what the movies and TV will say. And it is, I think, a hotbed of large pharma, certainly for many years. Born and raised there, went to Princeton. I'm expecting booze. No, we're at Yale. Okay. But I've been always interested in the intersection of different scientific disciplines. So ended up doing degrees in chemical engineering, bioengineering, and chemistry. And that was on purpose. That was to intersect between the engineering and the physics, the biology, and the chemistry worlds. And then sort of parlayed that into big pharma. So I've been, I don't know if I'm a glutton for punishment, but I've been in big pharma my entire career. I love big pharma. I love the complexities around it. I love the ethical considerations, the impact that you can have at scale, the challenges from both a scientific and a people perspective. It's a very difficult industry to be successful in the long term, but I think it's a really important one in terms of improving human health. So along those lines, again, being interested at the interfaces of disciplines and thinking that that's really a space where you could make critical breakthroughs that really transform the way that we operate. I kind of walked my way around the pharma industry, starting at Merck, in terms of what I call the 4 quadrants of R&D. So drug discovery, drug development, large molecule, and small molecule. And I've spent about equal space in each of those areas, ultimately leading to the role that I'm in now, or I get to play in all of those areas. But that's really what's driven sort of me is kind of moving to the areas where I think I can uniquely have the biggest impact in helping address issues in patients' lives.
Excellent. And so in your roles in Merck and at Janssen and now at Sanofi, you were a medicinal chemist, I believe. You've also worked in biologics. What would you say about the changing role that these specific careers have played in terms of drug discovery and development over the years?
Yeah, I think that what has been most impactful and important for me is when you practically experience making molecules with your own hands in the lab, when you practically experience developing commercial manufacturing routes and then going to a site to deploy them, when you do structure-guided drug design, when you work in biologics space, you get to see the challenges firsthand. So it's not a theoretical, it's not drug discovery takes too long, it's too slow, therefore we're going to disrupt it by doing this. It's a practical, this is what it takes today. We can't stop doing it today. Patients are waiting. But what can we do to make that future brighter? What can we do to make the future more effective? And so it kind of tempers the over-enthusiasm and hype that can sometimes come with new technologies, with the practical, how do you deliver today? But it really brings home to you, anything we can do in this space to accelerate drug design, for example, or to make a lower cost manufacturing route, you feel it because you've done it, you've actually done it before. So I think that that's something that's been very helpful to me in thinking about how to adopt and deploy and invent new technologies because of the experience of actually having to do it with your own 2 hands. A lot of my colleagues in chemical engineering will say inherently we're very lazy. We're always trying to come up with ways to not do whatever we're doing, to do whatever we're doing faster. So there's part of that that's true. It's an insatiable curiosity to do something better.
This takes me very nicely to some of the challenges that you may have faced in terms of bringing in innovation in big pharma and the different stakeholders that are involved in allowing you to realize the potential impact that these innovative technologies can have on the whole drug discovery value chain, from the technology to the people, and then the partnerships. What are some of the challenges that you've experienced so far, and how do you mitigate them? How do you balance all of these different roles?
Yeah, so the challenges usually stem from 2 areas. One is you want to bring together folks of diverse background and experience academically. And when you've been trained in different ways, you end up having different ways that you look at problems. And some of it's innate, and it's what draws you to a field, and some of it is trained. In certain academic disciplines, they train you to operate as an independent individual. In others, they train you to operate as a team. Yep. And so that's one challenge, is bringing together what the necessary diverse backgrounds to deliver on anything we do in a way where everyone can get on board and on the same page. I'll talk about that a little bit more in a second. The other challenge is then bringing together in a large company, which is complex and has all of different objectives within each different area. How do you make sure all of those objectives are aligned toward a common goal? And anyone who works in a large company will know that this is a big challenge to bring all that together and align toward a common goal, because my personal KPIs may not align with your personal KPIs, and that's a problem because the company's overall KPIs are supposed to be what we're marching toward. Yeah. So the way that I found that you can help with that, One is to work in different areas and actually spend time in those areas and get to experience, the life and the scientists from those different places. But you can't do that in every case. You need to be a subject matter expert first, and then span out and be sort of a T-shaped individual, right? I-shaped very deep, and then span out a little bit to be T-shaped. But you can't do that in every area. So the other piece that you need to do is go talk to folks and listen a lot. And I'll give a quick example from previous life way back at Merck when my team was developing a new process for Januvia. Sitagliptin was the biggest drug at the time before Keytruda came along. And we had invented a biocatalytic route to that process that was superior in every way technically. It was more robust. It was far less expensive. You could increase access to more regions by making the drug more cheaply. And it was very difficult in the beginning to get that adopted. And the reason for that, I sat there and wondering, like, why aren't we jumping on this? Why isn't this a thing that we're moving to immediately? And what I found was I needed to go talk to the commercial teams, and I needed to go speak to them. And I sat and I just said, after a while being frustrated, I said, can you just explain to me what the problem is? I want to hear from your perspective why you think this is a good or bad thing to do. And I just sat and listened. And I went from like convincing mode to listening mode. And what I found was they were seeing not the savings and the benefit. They were seeing all risk. They're saying, so you want to save hundreds of millions a year, but you're putting at risk a $6 billion product a year. And I said, oh, now I get it. So I needed to go back and revise and show how we could reduce the number of discarded batches, we should secure supply for regions around the world, and put it in terms that made more sense for what they were trying to secure. And we eventually did end up revising and adjusting and deploying that process globally. So it's a lot about listening and understanding the needs of the different functions around you.
Perspective, I guess, getting other people's perspectives, which is very great. And you have these wonderful tools, the snackable AI, the expert AI, and generative AI. How do you convince people to use these tools on a daily basis? I mean, in my experience in the last 15 years, getting people to use computational software, there's always been that barrier to usage, and you're deploying these tools company-wide. How do you get that adoption? How do you get into the brains of each of the different stakeholders and convince them to use these technologies? Yeah,
so there's the carrot and the stick, and you use both. The carrot is you show and demonstrate that it helps them in their daily jobs. Yeah. Because if you see your peer sitting next to you using something and they're able to proceed at a pace that you're not, then you're going to adopt it. Ultimately, fundamentally, you want to adopt latest technology. And I want to, if I may, take a minute to talk about that in a second and get back to that. But the other piece is, sometimes there's a bit of a stick, and you basically say, you know what, we're going to try not using PowerPoint anymore for our presentations, and we're going to use the Play app. And then people say, okay, well, I guess I have to learn how to use it. So there's a little bit of both. But if it's not superior, it won't get adopted. We are an industry, and scientists are a group of individuals that, we can be stubborn sometimes with our opinions, but we will adopt what's superior. And along those lines, I'll just mention that one of the things that I do love about the industry, sometimes we think it's a big behemoth, it's hard to move, and at times it is. But if you think about the industry overall, there are very few industries. If you look at the top 50 companies on the S&P 500, there are very few industries where some of the same names have been there, and some of those names are over 150 years old. The pharma industry is one of them. We've had the same names since the late 1800s. And why is that? It's not by accident. It's because we're an industry that does adopt radical change. Sometimes it's a little slow, but we do adopt it. And examples I can give are, when we started as a modern biopharmaceutical industry was probably in the late 1800s, when we went from just applying medical treatments that were from nature to saying, you know what, in nature there's compounds we can isolate, purify, and formulate, and understand that this is the compound that's having the pharmacological effect. But then the revolution of synthetic chemistry came along, and it completely opened the world to be able to design molecules that had never existed in the history of the universe for the express purpose of being a drug. So we adopted that wholeheartedly, or we would have disappeared. The computer age, the genetics age, and now the AI age, it's the same thing. We will always adopt the tools that can enable us to deliver on that mission for patients better.
Which leads me to my next question. So we've talked about the challenges associated with the people internally. The next is the tech. When do you decide, or how do you decide, to develop the technology in-house using your own expertise, or bringing in, say, data scientists versus partnering or licensing out?
Yeah, that's a great question. And it's going to depend on each technology. The way that I've approached this over the years is I don't care where the innovation comes from. If it helps us, we should access it, whether that's build, buy, partner, acquire, whatever it is. And the reason for that is our ultimate goal is to deliver life-saving and improving medicines to patients. So however we can access that more quickly and serve our patients faster is what we should do. And let's be honest, there's always more innovation that's occurring outside your company than inside. Just sheer numbers, that is the case. So the concept that not invented here is one that is not in my lexicon. So how do you get people to adopt it, and how do you decide when? What I like to do is take a look at what are we uniquely good at, what tools or capabilities or information do we uniquely have compared to a partner. And so I mentioned the collaboration with Biomap, but in general, it's true with many collaborations. Sometimes a partner has developed a unique capability, they've poured a lot of money and time and effort into it, and they've built a talent base up that's uniquely suited to deliver. However, they're missing the data. And we have the data, but we don't have that capability and army of people to build the tool. So that's a great idea for collaboration. Yes, we could say we're going to build it in-house, but then we're going to be 3 years behind whatever the latest technology is by the time we finish building it in-house. So sometimes that factors in is the time you can deliver and the tools that you have at your disposal today versus tomorrow. And sometimes you do want to build it in-house or over time be able to deploy it internally because you do see it as a particular competitive advantage. So you take it as a case-by-case basis. But I think that what you're really looking at is what are you uniquely good at and what does someone else bring that they're uniquely good at? And that's how you decide whether or not to partner.
That's a very smart way of thinking about things. And I think it offers you a lot of opportunities and brings in balance. Again, it's a challenge in terms of managing all of it. But I think that's very impressive. All right, so in terms of, I'm going to go back to people for a second, the computational chemists at Sanofi. Do you feel like their role is getting more and more important within the drug discovery and development space, or do you think it's shrinking? How do you think this role is evolving?
Yeah, I think that particularly in computational chemistry, it is getting ever more important. Their digital counterparts, there's a lot more support required from true digital people who have no experience necessarily in drug discovery in support of folks like computational chemists or computational biologists or protein engineers. And so that's also a role that's getting more and more critical, more and more important, where we have to staff up more and more people because the infrastructure and underlying base of data curation, data movement in your company, overlays of ability to pull in and out of different models and tools, that's becoming more and more critical and more important. So all of those fields are getting more important, and the ability for folks to interact and talk together and understand each other, at least on a base level, is getting more important as well.
All right. Okay, final question in this section. The pharmaceutical industry, 20, 30 years from now, do you feel like it's going to be more tech or less tech? Is there any turning back? Yeah.
I mean, I always feel like it's going to be more tech.
Okay.
So then the question is, what does that tech look like?
Yeah.
And what I would hope to see is that we can accelerate more dramatically by using, it's interesting. There are some cases where you need to generate more data in order to build models.
Yeah.
There are some cases where you have a ton of data. It's just so dispersed and so diffuse that it's hard to pull together to figure out how to build those models. We have both of those. We have a challenge in both of those spaces. So I see getting certainly more tech, and that technology is going to be a mix of AI and digital tools. It's going to be a mix of automation, so porting to the physical space, what's happening in the AI. I do believe that, I say always, so let's say within my lifetime, I do believe that we will always have to synthesize and make and test things. The goal is to have to be able to do that less in order to move faster. And the goal is also to be able to do it with higher quality. Yep. So the molecules we're working on now are far more complex. If you look at the structure of aspirin and you look at the structure of drugs, I mean, I'll give a biologic example out there, a CAR T cellular therapy, or one we're working on now, an in vivo CAR T delivering mRNA to transform a CAR T in the body. This is a living, autocatalytic, self-replicating machine that goes after cancer cells, right? That is far more complex than aspirin. So in order to deal with these stubborn timelines of discovery and development, where they've been stubbornly about the same amount of time over many, many decades, we're running just to stand still because the complexity of the disease we're going after and the drugs we need to make to go after them is getting so much more difficult. So I think that anything we can do to accelerate that, and a big part of that is adopting new technologies, both in the digital and in the automation space, is going to be critical to our industry.
Excellent. And it leads me to my final section, which is risk mitigation.
Yep.
We're going after more complex diseases where, disease biology is, as we're understanding it more and more, is extremely complex. We're designing drugs that are geared towards now personalized medicine. How do you manage that risk associated with the adoption of technology and in the decisions that are being made down the line?
Yeah, that's one that comes up a lot. So we hear about risks in AI tools. I talked about the explainable AI, and we'll talk about that in a second. The bias in the system that can occur just based on the information ingesting that is biased.
Yep.
So the way that we need to mitigate that is a couple of things. One is the decision and the responsibility always has to lie with the human being. That's the final ultimate responsibility is with the human being, and the decision is with the human being. These tools are used to inform and provide a guide for little pokes to say, hey, maybe you should look at this. Hey, maybe you should look at this, to really prompt the conversation in real time. On top of that, the explainable AI piece is important. It may not be important in every space, actually, but in our space, it is important. We need to understand why these suggestions are being made in order to learn from it, to do better, and to learn just as humanity. Why are we making these decisions? Why are we developing tools in this way? Why is that molecule being changed in that way? We need to be able to explain this to regulatory authorities. So ultimately, the decision and the responsibility lies with the human being, and that needs to be fixed throughout the process. Then with respect to risk about bias and things like this, I think that just as we have those risks today with human decisions, we need to be aware that developing AI tools doesn't necessarily get rid of those risks. So we need to continue to stay on top of that in the same way that we need to stay on top of it with our human decisions. So that's going to require, as I said, a level of understanding of what's going on with the AI tools, why it's making certain predictions that we need to build into the system. It's also going to have check and balances to ensure that the decisions we're making are aligned with our values and our goals, and that we can explain to regulatory authorities and patients why a drug does what it does, why we put it into development, why we want to put it on the market in the same way that we do now.
So you've spoken about the bias that could be introduced in terms of decision making, but what about the bias that's introduced in terms of the data that you're using to train any of the predictive models? Yeah. How are you ensuring that there is a balance in terms of the samples that are used to produce the data, et cetera?
Yeah, so there are ways. If I give specific examples about drug design, what you'll find is certain AI models are very good within certain species, within certain regions of structure-activity relationships. And the reason is that there's a lot of data. So kinases, a lot of folks developing drugs in kinases because that's where the data is. So it makes sense. It's a good place to start. It's a good place to go. But when you start to get to targets where there's less information, those models start to fall down a little bit. So what we can do in that space is a couple of things. One is actually generate data in the gaps. This is where automation comes in. Can we generate data in the spaces where we're data sparse in order to round out those models and make them less biased? In order to do that, we do need to understand how effective a model is across a range of areas. In the example I've shown, across a range of structure-activity relationship space. But I think that concept can be applied to other spaces as well, to understand and know where are we data sparse, to understand and know that the model is going to be highly predictive or less highly predictive, asking and answering certain questions, and therefore we might need to fill in those gaps with additional data sources. And some of that data can be generated intentionally in the lab. Some of it can also be generated with synthetic data. If we have really, really good physics-based models, we can generate some of that data synthetically. So I think it's about filling in the gaps and just being aware of where the biases will be based on our current data sets.
That's very cool. And what about in terms of clinical site identification or patient stratification and avoiding the bias in those decisions? Yeah,
so I actually think that the tools that we're having now, at least from what we're seeing, they're helping us avoid some bias because you're taking it out of the hands of an individual clinical investigator, not in terms of the decision out of the hands, but you're taking the only information they're using as what's in their head and their experience, and you're adding on top of it the experiences of every clinical trial that we've run, the experiences of all investigators. So although the goal is to eliminate all bias, I think that this is a step in the direction to eliminate a lot of bias because you're pulling in many more people's diverse experiences in those selections, and you're being data-driven data-driven. You're able to back up and say, well, why am I selecting this site? Let's say because this site has been able to deliver on the diverse talent or the diverse patient pools that we wanted in the past. This site's been able to enroll patients at pace. This site's been able to do things with high quality without making errors or mistakes. So you're able to do that in a much more profound way with a lot more information. And I think that will help get rid of some of the bias in the system.
Well, thank you very much.
Thank you.
So I'd like to take the last couple of minutes to open the floor to the audience. I mean, Sanofi is such a good news story in terms of the acceptance and adoption of AI, and then the outcome. And I'm sure many of you in the audience have a lot of questions for Matt. So I'm happy to entertain some of the questions from the audience.
Hello. My name is Li Juan Wan from Alexion AstraZeneca Rare Disease Unit. So it's so exciting to see how AI can accelerate the drug discovery process. My question is, do you have some idea or objective how many years or what percentage of time you can save for drug discovery timeline for both small molecules and the biologics? And do you see difference for the projection in different therapeutic areas, such as rare disease, oncology, or either? Thank you.
Sure. Thank you so much for the question. And it gets to the heart of what I know you care about and many others, about how do we go faster and deliver patients to medicines. So we do have certain targets and goals, and they are based not on, let's say, just fantasy of writing a number down, but based on an analysis of the entire workflow from beginning to end. Our goal we have a 3-year time horizon. My mind can't think beyond 3 years. I need to have things delivered in 3 years. Otherwise, I start to get antsy. So the 3-year time horizon, our goal is to reduce our small molecule discovery timelines on average across all therapeutic areas by 25%. So right now, our average is 4 years. We're fairly average in the industry for small molecules, down to 3 years. And our goal for large molecules, which were in the upper quartile of performance in terms of speed in the industry, we perform a bit better there. But our goal still there is to cut a year off of those timelines, which are about 3.5 years now. So that would be maybe a 30% decrease in the overall time it takes. That is based on an assessment of the drug discovery portion we're working on today, because as you know, there are many other pieces that go into those timelines. There's tox studies, there are stability studies, there are things that just take a lot of time. Now, hopefully we'll be able to develop tools in the future that are predictive enough that will be accepted by regulatory agencies so we can cut even more time off. But right now, that's what we're thinking in terms of average timelines. Those are our targets. With respect to the question about does it differ from rare disease, immunoscience, oncology, it does. We've found that in rare disease, and we have a fantastic rare disease area, a fantastic genomic medicines unit, we can go incredibly fast. We've had molecules move from, we call it milestone 0, which is portfolio entry of the target, through to preclinical candidate in under 2 years, several times over the last couple of years. So we can move quite a bit faster in that space. Because we have certain platforms where we can, lipid nanoparticle platforms, we can insert the gene of interest and move very, very quickly in animal models, and then ultimately go into human trials. So there are some spaces where we think we can accelerate more rapidly, but overall timelines, that's kind of what we're targeting. I would say in immunoscience and oncology, in the drug discovery portion, I see those as about equal. From what I've seen, the breakdown of relative timelines, they're pretty much equivalent. When you get into the clinical space, they can be different, depending on the biomarkers you have, depending on how long you need to run a study and the specific area that you're focusing on. But in terms of the drug discovery process, they're about equivalent. Yeah.
Hey, Mark, great talk, and thanks for letting us all kind of eavesdrop on your conversation with our friend from Canada. You just described a lot of efficiencies in the entire process on this drug development. Do you feel like the regulatory agencies, and in particular the FDA, are going to be able to keep up with this onslaught of potential drugs from Sanofi and arguably the rest of the industry? Yeah,
so it's an excellent question. And I think that if we work with regulatory agencies and we work together as an industry as we develop these tools, particularly the tools that relate to how we're doing filings, the information that ends up in the filing. So there's a lot of what I showed that's in the space that's preclinical work, right? And so that's a little bit easier to adopt because you then still have to go through all the clinical information and tox studies, and that's all very heavily regulated, and that all still has to be done in a let's say the output has to be in a way that's accepted by regulatory agencies and is understood by regulatory agencies. So some of the earlier work in the preclinical space I described is fairly easy to adopt. In the later stage spaces, we're going to have to work together with regulatory agencies, and I think the best way to do that is cross-industry, because I don't think that's I think that's a pre-competitive space for us, not a competitive space. For all of us to be able to work with regulatory agencies to make their job easier and our job easier in terms of filing, submission, and ultimately approval, it only helps everyone. But I do think that if we all go off on our own and do this in different ways, that could be a challenge, that could be a problem. Yeah.
Okay, so I have a question here. Thank you. So very impressive. Describe the whole process from targeting and all the way to FDA like a submission. The question I have, with this kind of tool available to you and the readiness of producing some kind of outcome to guide you to the next work, and how do you pick, like for example, when you generate a report, submit to FDA, it's so easy to generate the report in 10 minutes, and each report, like you run again, it could come up with a slightly difference. And sometimes it have a very important impact on the outcome. For example, go back to the selection of the clinical site selection. Some site might be omitted on the next one. And how do you decide which one would be the best for you to use? And how do you make sure consistently apply that kind of approach in your evaluation? So that's my question.
Yeah, that's a great question. And you're absolutely right that you can run that report several times and each time it'll come up slightly different. That's the nature of generative AI. That's one of the challenges. And that's why I put up there that we have a 70% final report. And it is, we say 70% final as in, from a technical perspective, it's 70% complete. It still requires 30% time for an expert writer to go in and modify and adjust it to be perfect. And that's something that we hope to get better, but our target is somewhere around that 80% range. I still think that for at least the time being, for now, it's going to require technical experts to still come in and refine, correct any of those missteps in terms of any missing information, make sure that everything's formatted correctly, what the agency expects. And so those clinical study report writers will still have a big job to do. And with our 50% increase in clinical trials we anticipate to run, this is going to help them to be able to take on that workload. But we have to be very careful to make sure that everything is precise and accurate. So in some ways, you can imagine a higher level of accuracy because it's directly transcribing from source information and populating the document. But in other ways, there may be things that are missed that you need that human element to come in and fix, which is where the other 30% comes in. So our goal in the beginning is really to say, can we shorten the process? Not can we displace human from the process. It's really, can we shorten it and pull it together for folks in a more profound way? And we're seeing the same thing in sort of knowledge generation and research, which I didn't talk about, but is the next thing for us. The time it takes a scientist to collect and collate information from publications, patents, internal electronic laboratory notebooks, it can literally take days or weeks to kick off a project. We have some pilots where we can do that in minutes. Now, will it be completely all complete and include everything you might want to look for? Probably not. But does minutes beat weeks and then you can fill in the rest? It does. So it's really a stepwise approach with a true understanding of where could the gaps be. And you've identified some gaps that are clear, that we understand, that we know are there, and that we have a human being come in to address.
All right, we're a little bit over time. I'm sorry. We have to stop right here, but let's all thank Matt for his amazing insights and information. And I'd like to thank you all for joining us, and please continue to enjoy the rest of the summit.
Thank you so much. Thanks, Patrina.
Thank you.